Hi everybody, this week we are going to study about the fluorescence. I chose several key concepts and key references from the Wikipedia. And in this lecture, first of all, we are going to study what is luminescence, and then in comparison, what is fluorescence. And then, how to use fluorescence for bioimaging. Then, the specific visualization for one protein. And then, fluorescent protein and its application. And finally, flow cytometry and its unique applications. Okay, let's start with the fluorescence. Here is one problem for you. In this picture, there are two apples hidden. Can you find those apples? Okay, one apple here and the other apple here. I just draw in myself. So even though you didn't find the apples, it may not be your fault. Important point here is, this picture is a black and white picture. If you want to find some object, you need to rely on intensity and contrast. Only those two informations. So that's why sometimes it's not easy to find your target of the molecules. How about this apple? It's much easier to find, isn't it? Why is that easier? Maybe you say it has a color, it's a red color. Then next question is, what is the color scientifically? Well, sounds like an easy question and may not be easy to answer in scientific terms. Color recognition is the ability to recognize different wavelengths among visible light. Then let's think, what is visible light? Visible light is one kind of electromagnetic wave, and it has quite narrow windows and about 400 nanometer to 700 nanometer range of short range of electromagnetic wave we call visible light. If the wavelength of the electromagnetic wave is getting shorter, then energy is getting higher, and if it gets longer, then energy is also become lower. And right outside of those visible light, shorter one we call ultraviolet or UV, and longer one we call infrared. To this direction, ultraviolet and getting the shorter X-ray and gamma ray. If it gets longer from infrared IR into microwave and radio wave directions. Now, very small range of this electromagnetic wave, the light we can detect, we arbitrarily call this is visible light, visible to human eyes. So this light is an energy form of photon, and they have different wavelengths, and our eyes has different receptor for different wavelengths. These three receptors we call RGB receptor, so red, green, blue. Now if you look at this spectrum, if you design RGB, would you design like this way? Looks a little strange. Blue is quite unique, but red and green is quite close to each other. If you design the receptor maybe in the computer system, RGB color system, you may want to make this system a little more distinct, isn't it? It may mean something. Now in our eyes, we have different light receptors. Some cells, like rod cells, a lot of rod cells, they detect the intensity and they detect the photons regardless of different wavelengths. So that's why these rod cells can help us to detect very sensitively small light in dark kind of things. Now, small populations of cone cells, those cells are responsible for this color the sensing, and they have different receptors for each cells, these RGB colors. Because of these color sensors, we can see different colors. Now, in this kind of a bullfighting, this fighter, he stimulated this bull using these red colors. Red color is similar to the blood and maybe stimulate people's emotion. But funny thing is, we can see three colors, but not the bull. Bull cannot see red color. Maybe it looks like to bull. It's kind of like this. So that means this red color is not stimulating the bull, stimulating people watching these games. It's a funny situation, isn't it? But it's a little exaggerating. Bull can see colors, but not like three colors as we see. Human eyes, we see three colors: red, green, blue, three colors. But bulls or the dogs, they can see only two colors, and they see a little different way. They have a blue, but they have a yellow. Now, this red and green, do you remember? They were very close to each other, and then dogs and bulls, they have only in the middle of this. So that means most likely this color, maybe they came from originally, they were in the same receptor, and maybe in the evolution of the process, they were divided into two different colors, maybe red and green. It's a possible story. That's why in human being, we have some kind of a color blindness problem, but majority of them is kind of either green or red problem rather than blue. Blue blindness is quite rare, but this the green and red is quite common. That's why to the blind, colorblind people, this beautiful picture looks like this. The green and the red looks like quite similar to each other. It's quite boring picture, isn't it? So we should appreciate the ability of the three color the recognition in human eyes. Now let's come back to this the red apple. This red apple, because of the color, it may be easier for us to detect for human eyes because we can detect different colors. But at night, even though there is apple still in the same site, now it's much more difficult to see those apples. Why is it? That's because of the property of the colors. Usual color we see, those colors are, it's a subtractive color. From the sun, all the mixtures of visible light coming through, and those mixed color as a background we call white light, and some part of the light. So for example, in this case, blue and green color was observed by the apple, only red color was reflected. That's why this color is shown to us as red color. Key point is, we need to have input light, and then some colors are removed, subtracted, and only reflected color we can see. That's why this color we call subtractive color. That means without input, there is no output of the colors. So that's why the color material is quite useful for us, but at night it doesn't work. In this case, if the apple can emit light itself, now this is a different story. In the dark background, if one material can emit light, emit photons, then this phenomena we call luminescence. If any material is luminescent, even though it's a single photon or a single molecule, that luminescence signal is so strong we can catch. So that's why luminescence can be much more sensitive than just the color sensing. This phenomena of luminescence. Luminescence is generating photons, and photon is a highly condensed energy form. If we want to get out some energy, we need to give the energies into the materials. So depending on those input the energy, we can classify this luminescence into different forms. First one is the bioluminescence. Sounds like we can utilize biological energy. What can be? It's ATP. Utilizing ATP, we can generate the light. Or you can utilize chemical energy. Highly condensed chemical energy can transform into light forms. Or we can utilize the electron or the electricity to generate light. And then we think about the fluorescence. Examples of bioluminescence. This firefly can be quite famous one. In the dark, we can see this light is coming through, and this light is generated by utilizing biological energy. Deep in the sea, there's some fishes. They can generate this kind of light utilizing again bioenergy. So this is also another example of a bioluminescence. How about the chemiluminescence? Glow stick. So inside here, there is some chemicals. They can convert this chemical energy into the light forms. Now let's look at the chemistry of this glow stick. There is this highly energetic materials inside. When they meet with hydrogen peroxide, and then they can generate this kind of activating molecule. This activating molecule can activate the dye molecules. This kind of molecule we call sensitizer. This sensitizer molecule can activate the dye into the activated state, and then eventually this dye coming back to the ground state and generates the light. So this is the one example of chemiluminescence the mechanism. And how to make the glow stick, we can think. Maybe outside here we can put dyes and this material together, and in this case hydrogen peroxide is the catalyst to initiate these reactions. So in this glow stick there is a smaller tube, maybe made by the glass, and then if you just bend the glow stick, then break glass tube, then hydrogen peroxide can be released to this solution and then initiate chemical reactions. And they can spread out the whole part of glow stick, and then it can go minutes to hours. Now this is chemiluminescence. How about the electroluminescence? Actually, we have a lot of the electric bulbs in our life. Those are all of them is kind of electroluminescence. Recently, more and more of this LED, light emitting diode, is becoming popular because of their high efficiency of transferring electric energy into light energies. So as you can see here, by providing this electricity and then change that energy into light, this is electroluminescence. Then now let's think, what is a fluorescence? In the daylight, it looks like it's a white color, but under UV light, now it emits specific colors, in this case like a reddish colors. That means this molecule can absorb some light and then convert that light different wavelengths. The input energy is a light form and output is also light. We call this process as a photoluminescence or fluorescence. Many stones found in nature, they have this kind of fluorescent molecules inside. So depending on different colors, it can show all different color here. As you can see here, the background is quite dark. That means input light, UV light. UV is not the only source we can utilize, but UV is convenient for our eyes because our eyes cannot detect UV. So that's why if you utilize UV for excitation, the background is very low and we can just see only those colorful parts to our eyes. Not only this kind of the non-life forms of stones, also there are a lot of fluorescent life that has been discovered. So fluorescence seems like it's quite a common phenomena in our nature. The first step of this fluorescence is the material somehow should absorb the light. This is the input energy. And when it is activating happens by taking off the light, and then electron can jump into the excited state. And that electron coming down a little by this non-radiative degradation, and then they are accumulated here. Regardless of those original excited positions, all of them, they are collected at this stage, and then they can emit light. Now, if you look at this the two diagram, excitation or absorption and emission as a fluorescence. Now, energy-wise, excitation is bigger than emission. That's why usually the absorptions happens in a shorter wavelengths and emission becomes a little longer wavelengths. And those difference, the peak to peak, absorption peak to emission peak, this difference we call Stokes shift. If the Stokes shift is bigger, that means that this energy loss is bigger, then emission can be much longer wavelengths than absorptions. So this is a common mechanism in fluorescence. If you look at those excitation spectrum, now this has different peaks here. Now the question may be, which one is considered as maximum excitation wavelengths? If you plot this into the fluorescence, now this is absorption or excitation, and this is emission as a fluorescence. If you excite at different positions here, what happens? You remember in previous case, whatever the energy level you excited, it goes up higher, but they are collected in the same level and then emitted together. That's why, regardless of those excitation, you can get same shape of emissions, just those intensity can be different. Let me repeat again. Even though those excitation spectrum or absorption spectrum is quite different, and then you utilize a different wavelengths for the excitation, the emission shape is the same, only those intensity can be changed. If you think about the brightness of a fluorescence dyes, Maybe the first step you need to think is absorption. This is following the Beer's law. The absorption can be dependent on extinction coefficient, and this is unique parameter for different material. And this is bandpass, the length, how long the light can pass through. This is the unit. And C is the concentration. Among these three parameters, B and C is environmentally dependent parameter. Only epsilon, extinction coefficient, is unique parameter for the material. Then brightness can be defined by this absorption amount depending on this extinction coefficient, and then quantum yield. Quantum yield is per introduced input light, how much light can be generated. That ratio is a quantum yield. As the first step is light absorption. Most of those dye molecules should have strong dipole moment, electron donating group and electron withdrawing group along line this the conjugation system. Using this strong dipole, they can absorb the light. Dye molecules, when they have a lot of flexible groups around, and then even though they can absorb the light, they can be strong, colorful material, they can lose their energy by vibration. Then, how to change this molecule into fluorescent? One trick is, by making this kind of a bridge here, then suppress those vibration or rotation, and this molecule becomes more rigid than original molecule. Then, eventually it can emit the light back. Now, if you look at those brightness and the wavelengths of different dyes, now we can see here. So this is the emission wavelengths. And if you look at those colors, let's say, we cannot see shorter than 400 nanometers. So these materials are kind of a no color material. But over here, starting from blue color, and then 520 of green color, and then yellow color and red color. Above the 700, it's difficult to see using our eyes. Now brightness, again, this can be defined by extinction coefficient and multiplied by quantum yield. This can define those brightness. And some molecules here, those are highly bright molecules. Fluorescent and body P here and tetramethylrhodamine, this kind of class of molecules are kind of representative, very bright dyes. Another very popular dyes are here, size 3, size 5, size 7. Even though they have relatively low quantum yield, but they have an extremely high extinction coefficient. They have been utilized very popularly in genomics and proteomics. Here are some examples of most popular fluorescence dyes. First of all, the blue dye, DEPI. DEPI has been used for DNA binding, and it is the staining dye for nucleus. And FISI, we just have seen those structure of a fluorescent, and fluorescent structure is connected to this NCS, can react with the amine group very quickly. So this is kind of a labeling reagent. And tetramethylrhodamine is a slightly longer wavelengths, like a yellow to orange color, and Texas red compounds. And compound name, FISI is a compound name, but now utilizing this compound name, even those filters for microscope, it is defined as a DEPI, FISI, and this TMR, tetramethylrhodamine with ITC together, TRIC, is the same name for this TMR. Now FISI, TRIC, Texas red, and those popular cyanine molecules, size 3, size 5, size 7 molecules. These molecules are most popular the fluorescence dyes, something you want to know the names. Now this is the introduction for fluorescence.